You have to, otherwise I can't hear myself. Can you hear me in the back there when I talk like this? Okay, I'll try to do this for 2 hours then. I've seen some of you during the introduction day. Well, from me again, welcome to the study. Oh, this is the first day in life that you know what to do, right? Will it be bioinformatics, or will it be systems biology, or will it be both to some extent, right? That's at least the idea. And if you have not thought about that problem, just study on until we perhaps clear it later. Okay, so today I'll, on the program there is Outlook. What is bioinformatics? What can we do? What are the current big questions? And so on. And I will try and argue that bioinformatics has become a data science. So, first point, modern life sciences are really information sciences. And who by now knows the first definition of bioinformatics as received by the inventor of the word bioinformatics, Heringa? Who knows this? It's very much in keeping with this statement. So, bioinformatics has been defined as the, well, day one, as the field that studies information systems, that studies informational processes in living systems. That was the definition that Heringa gave. And so that's in keeping with this. But another point is what is happening more and more over the last number of years or so, and it started in 1995, really, when the internet took off. So it became far more easy to work together. Till that time you could send an email, you could even get sometimes an email back. It's not so long ago that all of this still wasn't there. And people had to send each other letters. Now the next course, algorithms and systems analysis, I'll tell a few stories about really people sending letters around with ink and so on. But not now. So, this is the state of the art. And if you ask how did this all come about, why did it so much turn into an information science? Well, then the best thing to say, I guess, is that's because we can, since the last few decades, we can measure more than ever before of biosystems. Still, cells don't have plates like here is the Golgi and so on. We have to reverse engineer to try and understand it. But still, we can measure so many things, and all kinds of new measurement techniques are coming up. For example, what is now going very fast, the single cell measurements. Very important if you are studying a tissue and you think this is all one type. No. Each cell is its own identity. We can really do very different things. And you know why that is especially dramatic in, for example, cancer tissue. There, the heterogeneity is far greater than in normal tissue, healthy tissue. So that is something we could use, perhaps, but then we have to know about it. So these techniques are really very important. And now even single molecule tracking is coming up, where you can follow a protein, being conceived on the ribosome and then, go through a cell into all kinds of cell organelles and so on, or passing a cell barrier, a membrane into another cell. All of that stuff is happening right now. So that a lot of fascinating movies about the protein world might come up. You can see this happening if you can follow them individually. All right, so that's going to happen. But of course, very importantly, and that's also partly why you're here, it has all to do with computation. Data, it comes off a data generating machine, whatever it is, but then it has to be brought to life and made usable. I'll talk to some extent about that in this lecture. So IT, computation is really key to all those techniques. And most of the machines indeed nowadays have just a big computer connected to it because the computer is already there when the data is set. Okay, so all of this was happening, and so maybe the most important thing in history for bioinformatics, because we did this for decades, the first definition, let's say bioinformatics originated in 1970. So that's a while ago now, we always say it's a new science, completely new. Well, after 50 years in the meantime, that's a statement that it comes smooth. So we have to think about something else, but definitely in the meantime, 2 decades ago, here we go, 2 important papers arose. These are, if you write papers in these 2 journals, you'll be very happy as a scientist. These are maybe arguably the most important scientific journals, American Journal of Science and the British Journal of Nature. And in the same week, a paper about a very important thing, the human genome. The first time, really, measurement techniques, the human genome was measured. And the DNA molecule became known in molecular detail in the sense that we knew each of the nucleotides. Why was that? It sounds, two papers about the same in the two most important journals in the same week. That sounds like orchestration, and it was. And so what had happened is, this was a result of the Human Genome Project, and you know what? It was maybe 2-3 weeks before this press conference that the world in this era was still at war with one another, because there was a private player and there were the allies, so to speak. It's almost war terminology, but it was war in a sense about who would be the first to crack the human DNA. And I'll tell you a quick story about it. And that stayed really with the patenting issues, because and I'll show you his face. Yeah, I can do that here. You know that great gentleman in the front, I think. The question is, two presidents ago? That gentleman there that makes a bit of the impression as Darth Vader or something. And in a sense he was. There was a man, and I'll get to him. His name is Craig Venter, and I'll show you that here. Quick story about this man. He worked at NIH, National Institute of Health in America, very powerful, and in those days rich medical institution across the United States, concentrated for the most part close to Washington, D.C. And Craig went to his boss and he said, you know I'm a genius. The whole world should know I'm a genius. Okay, great, that's very good. Yes, you're clever. Thought about this the Human Genome Project. I've seeked a little bit of yeast, and I can pull this off. So why don't you give me 30 or 40 staff scientists? And I'll do this in my group, and everybody happy. And I'll do this for you, and you'll be very famous because of me. So the director said, Craig, yeah, that sounds all very sympathetic, but and there are more staff. What about 5 positions? And that's already, I would never get this done in this university. I tell you, 5 is amazing if you get it. Shall we talk about the potential position in 5 years? That sort of answer you would get. So that is quite something. But it wasn't enough for Craig Venter, and he said, you know what, you'll regret this, but I tell you now, I'll go out of this place. I'll fund a private company, and you know what, I already have some venture capital. I'll found a company called Celera, and I'll pull this off, and and now another thing, I will patent each and every gene that I will find, so that any medical research has to come to me first and ask me whether this can go ahead or not. And of course, at a nice price that I will set. So that was the threat. And then people thought, this is ridiculous, this guy, big mouth, it won't happen. But then, we started publishing papers about other genomes, yeast a bit further, and the quality was pretty good, and he had a new, very fast technique. What he also did was, he bought up a few of the most famous and cleverest computer scientists in the whole of the country, in the US. In no time, he had the biggest cluster computer, the most powerful computer in the whole private business worldwide, so he pulled a few things off already, so people got a bit nervous. Is he crazy, or what's going to happen? Will this nightmare scenario come? And then people say, yeah, but think about it, patenting your genome, what nonsense, your genome is yours, how can anybody else? So they thought, we look in the law and it will be clear that this can't happen, this is unethical, we feel that it's unethical. Then of course, the law was silent, there was nothing about it, because people in the older days had not thought about these type of things coming up. So then they decided, okay, if we can't win by law, and this guy is good, then we have to try and act and speed up and get it done earlier than this guy, otherwise, we have the patenting problem. So that was what happened, and Terry Lander will be on a picture later, which I know personally, the director of the Broad Institute, an important genomics institute in Boston in the US, was phoned one, I think it was a Tuesday morning, he told me, 9 o'clock, phone rang. Hello, that's what Americans say when they pick up the phone. Hello, this is Bill Clinton here. The guy would just say, oh, Mr. President, what can I do for you? I hear there's a problem in sequencing, and that it's important business people have told me. Could you use $50 million. Mr. President, I think I could use it. Yeah, why not? So that's more than 30 positions, of course. So they went ahead, and there were a number of chunks were single-handedly given out by Bill Clinton in that period. So at least this was sort of a sign that politics there knew something is happening, it's important, and we need to act. That made the day for bioinformatics. This was because then they said, who are people can deal with this information? Yes. So that was good. So genome sciences were married to bioinformatics. Since then, and the field has done well also. Since then, so that was a very important moment. So I'll flash a few just slides, past few. Great inventor, don't forget it. So he has been very important in the field. Right? Maybe sometimes for the wrong reasons, but at least he gave a presence of the field and so on, right or wrong. By the way, I don't think I have the slide. I'll first show you. This was a president who could speak beautiful phrases, Bill Clinton. And so here's one of them, most wondrous map. Here you see a nice party. This is Francis Collins. This is Great Venter. They were bitter enemies. This was the leader of the Allies, you might say, of the public research institution trying to seek us like hell. And this was the Darth Vader of the whole area. Doing all this private work. So the handshake that the current president hands out nowadays is nothing compared here. These guys were, trying to squeeze one another like hell. And here you see Clinton somewhere standing between the three of them. After this thing, I was trying to keep them apart because they wanted to kill each other. But it looked like a happy party. Another thing that's nearly funny, I think, is where is he? Yes, you see the UK wanted to be there. Should it become political? After Brexit, everything will be fine. Well, you get this, Anyway, it's not a political one. I used to live in London, that's why I have some feelings about that. Okay, that's the situation. But luckily, 2 weeks before this press conference, 26 June 2000, most wondrous map, the Allies were devastated. They said, we're not going to win. Venter is doing it. We have no way to build all the little chunks of information together into something that looks like a genome. We're lost, we've lost, this is, cataclysm, disaster. And so the one clever little guy, well not a little guy, but one clever person, made a crappy algorithm, but it did the job. And then Venter found out, oops, they can do it too. With another technique, and then they said, oops, now what should we do? Let's then pretend to be friends, and then we worked all together, right from the start. Some nice acting. But, in science, as in other parts of life, all is well. That ends well, and of course, more importantly now, we're all sorry for it, right? You can see that here, they're grinning. So that's the story. So, because of this pressure, technology, was developing like mad. Enormous steps have been taken. If you think about this project, you could compare this as, you want to change your bathroom or something, or build a bathroom in your house, and you call in an engineer, and you tell the guy, I thought about a nice power shower, and this guy says, well, what is a shower? I don't know a shower. You're supposed to, I build a bathroom, but you don't know bath, you have to explain the concept to me. So that's not very reassuring that an engineer like this could do it. And that was basically the state. We had no knowledge about the technology of building it together. And there was nothing also even to at high speed, measure these nucleotides. And so, it was amazing. And then they thought, in 1995, right, 5 years before the year 2000, this will take 10 to 15 years, and it will cost 10 to 15 billion dollars. There were some estimates made. So, in fact, it was pulled off in 5 years, half the time, at a cost of roughly $3 billion. That's a lot of money for a single genome, you might say, but it was the first one. Now it's a lot cheaper, as you all know. But, point taken, is half the time and 20% of the price. Hey, I want such an engineer for my bathroom. It never happens. It always takes 3 times as long and double the price or something that initially agreed, right? And this was a bathroom maker who didn't know about bathrooms. So, pretty impressive. If you really work together, what people can do together. Okay, since then, technology has been flying. It might be that you've seen this slide already. The Human Genome Project here is here. The Sanger technology. Sanger has had 2 Nobel Prizes. That's exceptional. Some people did that, but he got 2 Nobel Prizes for chemistry in the same field. He's the only one who has a Nobel Prize in the same field. So technology, but you see nowadays that the speed of technology going up and the price goes down with that. We talk about the $1,000 genome, but it will be $50 in not too long. And here you see some interesting developments with the read length. Of course, what we hope to do, you have a DNA molecule and in one go you measure the whole thing, all 3.3 billion letters. So far, this has been a really crude job. You would cut the... did you have a sequencing? No, not yet. A little bit? Okay, then I can sort of gloss over it. That's good. Very small little snippets. You need to calculate forever to try and paste that together into the book of life. The DNA molecule. Here you see that the length is, here is 10,000. In the meantime, the read length goes up to 1 million. That's not 3.3 billion yet, but still, we're getting there. So that's very good. And what is even more amazing, I think, these two pictures are not at scale. That machine is roughly has the height of a person. And this thing is a memory stick. That's the minion. That's the thing that does the sequencing. You could take it in your pocket to Africa, to go to an area where there's Ebola virus or something like that, and do the genomics there and then. Well, admittedly, you need a little suitcase with some further stuff to do the lab work, but really, you walk and you can do your work. So that's quite something. And these developments are going on. So, amazing stuff. Keep with this company. It's a new way of doing a company. It's not so much for profit, so it's really interesting. In all aspects, business model and what we're actually doing for the world and for science. So basically, you might say, if you think about a career in this field, you shouldn't be too faint-hearted. You should like to, like new developments, new breakthroughs, because that happens really all the time in this field. And so, new techniques to measure, of course, all the time as a result, new data, new formats. You thought you were onto something, you had established something. Next week, another guy comes about, and let's do it like this. Oh my God, all my ideas out of the window. You have to do that continuously. But what is very nice, I think, all the time, new insights. But then if you don't like this, I have another advice for you. Why don't you leave the room now and go study? It's not to be derogatory to people doing ancient Greek philosophy. Great and interesting, but the data is not generated at the speed that we do. Maybe an old book here and there will be dug up in some library, but data growth is not so much. And insights develop, but at a bit slower pace. Right? So, not all scientific fields develop at the same speed. So you can choose, what fits best to your personality. For example, with us, this is already an old slide, but very many years ago, and there will be this course where we do a lot of sequence alignment and sequence analysis. There were all kinds of pieces also in the human genome that we said, hey, this bit is very conserved through evolution. All kinds of animals have it. Even plants have these motifs. We can't find out what it does. From the course you've just had, you know already what is conserved is probably functionally important, right? So how can it be that there is such a fingerprint there? You can't find it. So, and then when you don't understand, when something is new, you try and give it a name, and when you don't understand it, it's really important to give it a very good name. So these spots, non-understood spots, were called hotspots. It sounds very nice. The problem was these are spots that are conserved and you have no clue about. It's a bit of a bummer, really. So then some 10 years or even more later, of course, you have to wait for it. Somebody finds out that these regions have an important function, but not at the protein level, but at the RNA level, because in those days RNA biology was born, and a lot of knowledge came about. RNA structure and RNA sequence. And another example is really important now, last 5 to 10 years, I guess, so relatively new, microRNAs, a whole family of small RNA molecules that have a very important function. They're really center stage in cancer, cancer science, cancer research. But they were not known all too long ago, so a lot of those hotspots are the places where microRNAs are encoded. Yeah. So we're measuring, and a lot is happening. Anybody is measuring in this world nowadays, not only in biology. Think about the internet, think about Spotify, think about all kinds of stuff. Facebook. It's huge data. It's really big data everywhere. And so you see that, and a lot of people, they don't know what to do anymore. I'll show you a few slides that talk about really, physical storage of data that becomes a problem. So just a few examples of other sectors of society where a lot of data comes about. And of course, the question all the time is how to convert all those data and understand it. Right? We want to do good to this world, or we want to understand biological systems, so we do want to do remediation, for example, if some individuals get ill, so treatment, knowing how to live a healthy life, we need to study. And so all this data should be used. And of course you might have seen a couple of those figures, in the bio business, biosciences data just typically grows exponential. That holds for the protein structure database, it holds for sequence databases, and it certainly holds for sequencing. To show you that here's an old slide, I must say, but this is on a logarithmic scale. It means a straight line is already exponential growth. And if you look some here, you see even that this exponential line, or straightened exponential line, even goes up. So it's almost super exponential growth, you might say. So this is for a number of structural and sequence databases. So that is going on. Growth is still exponential. Also, the scientific literature is growing exponentially. Right? And these speeds can all vary. Now, you have, of course, a number that is the index for the exponent, but everything, basically, sooner or later you'll run into a problem if you want to store all this stuff, if computers don't keep up with that. So that made a couple of people think about it, and so she's retired in the meantime. She's one of the important commissioners in Brussels for the EU, and she spoke words, data is the new oil, or data is the new gold even, and you see here, and yottabytes is sort of, if you add up all data produced per year on this planet, we're at, I think at the moment, at 10 yottabytes, 10 to the power 10, so this is 10 times 10 to the power 18 or something bytes. It's a lot of data, many zeros behind the one. So, welcome to the world of big data, you might say, and we have a lot of big data too. Show this. Let's show this one. So everybody talks about, getting out of hand. Big data is growing ever bigger, and what do we do with it? Well, let me show you something. There was a paper a couple of years ago in Nature, and that didn't argue that data is growing, but data evaporates at the same time. How can data evaporate? Here's the plot. This was supplementary data, accompanying scientific papers published in Nature, one of the top journals. And supplementary data in those days was stored with the scientists themselves. So they had a paper in Nature, and there was a link in the paper, and there you could check all the tables and all the details and whatnot. So fantastic. So here you see that after a couple of years, a quarter of the data is gone in really a few years, and if you wait long enough, all the data. That's it. So what is this? Data, of course, doesn't evaporate. Well, disks may break down, but there is not so much in the way we store data that it really, evaporates like liquid. But of course, data disappears because people throw it away, for example. Things break down, so that it's not stored in the proper way. And what typically happens, of course, when we talk about the paper, oops, that data was on the laptop of this, whatever, Moroccan student who went back to, or Chinese who went back far away to the home country, and took the data with him or her, and we don't know where the person is anymore. So sorry. Yeah, it shouldn't happen, but this happens a lot of times, right? Did it ever happen to you? You needed to get to some important data set and it was difficult to find it? Has that ever happened? No, of course not. I see all straight faces here. Very good. Great. Okay. Now you work on in bioinformatics, you'll get to this stage. But okay, I'll talk on this a little bit more. More is going to come. This is the data growth, of course, that I already showed you. But here's something else. I think some of you will know Moore's law. That's sort of a law of the growth of computation. It's much deeper than that. But let's say ballpark figure is computer speed. How fast we can calculate globally doubles every 18 months. But also the size of the disks, unified disk space over the globe also doubles every 1.5 years. Okay? Look at genomic speed, and this is a lie. 6 to 8 months is more like 5 months now, the doubling time. So see what happens. the disk grows like this, but genomics data, only the sequencing data grows at a much, much faster pace. So there will be a moment that even if you say, you know what, forget your Facebook guys, you won't mind, will you? We'll use all the storage for genomics data, and there will not be enough. What are we going to do? Stop all these measures? Or, so, the things you can do is, of course, is try and compress the data, you put it on, you know, that it's maximally economical. Another thing is, okay, now we have to start selecting. We can't store everything, so we should now decide what to store and what not to store. For example, what happens in genomics data a lot, you know the human DNA, 3.2 billion letters, store them cleverly, 3 gig. That's nothing, right? Maybe 6, depending on how you store, but let's say 3 gig, you can store a DNA molecule. The problem is, all the measurements you do, and then the imaging data you need to, you know, have the protocol of what you actually measure, that's easily terabytes. So that is a problem. So the people say, you know what, the primary data we'll just throw away. We measure the sequence, we can reconstruct the sequence by primary data, no space. And then later on there is something, hey, this nucleotide here, this region is not so clear. Shall we look how we measure that? Maybe there's a measuring error. We don't have the primary data anymore, so do we do? We measure the whole thing again, and then it's about, what do we know now? What do we need now? Brilliant, we need a DNA molecule. So that's how you get biobanking. So nowadays, biobanks, fridges full of DNA all over the world with software to make the DNA findable. You don't want to wait half a year to get the molecule again right, so this is all happening. Okay, so this is compressing. And now if I would ask you, think about, say if you want to store 10 or say in the meantime 50 yottabytes. How many disks will we need? Would it fit on this table, you think? Even more than this, would you say? Say no, please. I would say no. What about this room? Would it fit in this room? Would it fit in this building? Could we cover the whole of Amsterdam? Or could we cover the whole country? Not the whole country, it's a small country, but could we cover the country? No. Well, if you're uncertain, I'll give you another suggestion. There is one molecule that is roughly 4.2 billion years old. It has a 3-letter name. You might think of some molecule, right? We'll talk about it a bit more. And then, you know what that capacity is. You know how much you need for 1 yottabyte, roughly 1 gram of DNA. So much. Maybe. So that's amazing. And DNA is very stable. It's maybe more stable than a disk. You know, we have mammoth genes, and we believe that, that has been in permafrost, and that's still, it's a very stable molecule. So this is an amazingly efficient molecule when it comes to storing information. That's what DNA is doing. So amazing molecule, really. Respect DNA, I would say. It's really fantastic. And so, what can we do in the meantime? All the sequencing projects have shown us that we can read out DNA. Maybe for use like disks, it's not fast enough, but that we can do. The problem, of course, is how do we write a disk if you use DNA? How do we store information? You have to be able to make DNA molecules. That's not so easy, although there are all kinds, and George Church is one of the champions, Harvard professor. He's really a genius. I'm not sure about Craig Venter. He's very good. This guy is a genius, I think. So he has published a book, and he's put the PDF of the book in DNA. So he gives his book out as a DNA molecule. People can't do so much with it, but anyway, it's enough to Okay, so once again, what will happen with DNA technology or storage or computation? The planet will go on changing as a result of all of this technology and information technology. And we'll see a lot of stuff happening at the moment, of course. Right or wrong, but it's shifting. So in science, or big questions, the Higgs boson was being discovered in a sense a few years ago, thanks to big computing, being able to follow the data and all these particles, large-scale surveillance, right or wrong, I said, AI technology, you hear about China, and imaging and face recognition, and in many countries that happens. Of course, long-term weather predictions. Some people, some political figureheads think they can do it off the top of their head, I understood recently, but normally we need a lot of computation to know where all these high winds go and so on. Landslides, India, big problem, for example. Spread of disease. A few years ago, Google had a nice experiment. You know, there are in the world, there are a few expert places, one of which was the lab I was at in London at the time, National Institute for Medical Research. There are a few in Asia. The flu originates every year in Asia and goes westward, so they can sort of trace it and follow it. And every year for the flu jab, they're looking, oh, what should we build? And it's a gamble, and sometimes one year the jab's better than other years. Guess what? The spread of flu was studied by Google, and they just used their email. People saying, oh, you know, I had a cough, I didn't feel so well, they just traced that. It's not, you know, they don't need clever technology. They really see how the flu spread much better than people who try and do it on the viral content of flu, right? And some social trends. Now there are much nicer examples. It was something that my computer science colleagues liked a lot. Oh, do you see the Spotify was used to, to relocate a rap concert? Fantastic. I'm sure a lot more interesting stuff is happening based on big data. Jay-Z, is he still the man? Yeah? Okay, at least that. Good. All right. So, concentrating a bit on data. So, I've started alluding to that already. There's a hell of a lot of data out there. Most of that is a mess. If it's not forgotten on some disk, it's stored in a way that doesn't make it easily usable. Now I'm saying very kind words. So basically, you know, the sources are incompatible, the measure. There is, of course, no biologists are the champions in calling, you know, the same thing with 20 different names, or the other way around. Everything happens. Everything is ambiguous. So there are no naming conventions, or at least no standards. A lot of time, the data, it comes, and then, you know, the technology to look at it or to analyze it is not in keeping. I'll give you one personal story that shocked me. One of my best friends died in 2011 as a result of kidney cancer, and I was there on the way, and I went with him to all of the oncologists and so on. So it was in Utrecht, he had an operation, and they said, you're clean, you know. Nothing, your lungs are clean, everything's clean, you know, you're a few lucky ones, you can go on with your life. And I said, okay, Rian, you know, how do you feel? Let's go for a second opinion if that is really true. So we went to a colleague here in VMC, and he says, and we had the CDs with that, the data was on, and this guy takes the data, puts it in his viewers, and he says, what did they tell you in Utrecht? Yeah, I was clean. Well, I disappoint you. I see 3 metastases in your lungs, for example. And I'm not so sure, I think you need treatment. So, of course, he went back to Utrecht and said, guys, what did you do? You know, you said I'm clean, and I'm not. And then they said, it could be that we looked with the wrong software at the images. There we go. Just a little error. So that's state of the art in hospitals, right? Now we talk about, so it's not only the data problem, yeah? It's everything, software that uses data. We need standards, we need good things, because in this case it's life-threatening sometimes when you make errors at that level. Okay, so data is important, but also treatment of data. There we go, 2 slides on this. I love this statement, so that's why I flash it here to you, show it to you. You know, data is important. You know, people in the old days were massively clever. Maybe they used their brain sometimes in a less lazy way than we do. You know, they had to work everything out on paper, think everything through. We just use we use a model, and we let the computer tell us, oh, then, you know. And so it's another way of thinking. I don't say what is better, but certainly different. But they like the data. So this is really true. You know, even a genius like Archimedes couldn't do it because he didn't have the data. So data is really important, and you know, I started talking a little bit about that already, and I'll do a bit more to hammer this message home at least. If you forget everything of this later, I hope you will not forget these type of things, very important. The Dutch students here might recognize the thing on the bookshelves of the grandfather in the meantime, I guess. Anyway, you know, in the old days, if you had this was a very expensive dictionary, encyclopedia. And so, you know, well-respected families, or they thought they should be, bought that thing, put it in there on the bookshelves, never looked in it, of course. But anyway, if you came in and you saw that, ooh, this is a good family, you know. Let's bring the daughter in, and so on. And, yeah, so that's fantastic. And so, but that meant, you know, there were messages in there, and they were not, let's say, the quality of that information was better than the internet nowadays. Why is that? Because there was a lot of filtering here. Experts would write the text, editors would look over it, then it would sort of appear in a book like that. And you know, not too long ago, at least in the Dutch language, if you said, how do you know, come on, how do you know that stuff? What are you talking? And then you would say, oh, I read about this. Oh, okay, if you read about this, then obviously it must be true. Well, I guess that's history now, right? So what has happened, very good thing, access to information was in the old days an elite thing. Only the upper class would have access to information. Because there were filters, it was not for everybody. Thanks to the internet, really virtually everybody can get access to information. But now the burden is on all of us. How do we treat that information? How do we interpret it? How do we use it to our advantage? And there you see it's a market with dark and light corners, and we need to do something. So, and in science the pitch is, okay, public data, you know, science is done, it costs a lot of money. As a result, data is generated. I think there is an obligation, excuse me, to do well with that data, to make it accessible. And I show you a few of the technologies that have been developed in the remainder of this lecture. So, this is a philosophical question, how should we treat that data? I'll tell you another story. It's a bit about geniuses, I guess. This is another one. And I was really a genius, very clever, very successful MIT student, but young Stephen Keating. He was featuring in all the important fake, in the meantime, like Washington Post and New York Times, right? Journals in the US. And so what happened, let me tell the story. Should I make you read with it? No, I'll tell you the story. This guy was a neurobiologist, and he loved computation, and he loved 3D printing. Here, in his hand, is a 3D printed object, which he gave to his wider family for Christmas. You know what it is? It's a bit gruesome. It's straight. This was the tumor that was taken out of his brain after a 10 or 12 hour textbook on open brain surgery. What had happened with him? He was at MIT studying and liking all of this, and then there was sort of a new screening technology that was MRI based of the brain, and he said, you know, for the fun, I'd like, you know, find out how my brain looks, so let's join in the fun and let's join the group that is being measured as a test protocol. So that happened, and then they saw a small spot in his brain. And they said, you're young, we've seen this more often, maybe you'll rescan in 5 to 10 years, we're not worried at all. But if you hear that it's your brain, you get a bit worried, of course. So people thinking about that. And I think he had a rescan a few years later, earlier than they said. He went after 3 years or something. And then, you know, no changes. Okay, go lead your life. Don't worry about it. But then I think, again, 2 years or 1 year later, he started a few minutes per day. He had a very sort of a smell of vinegar. And he would ask, here's no. So he was doing it. And then it was pretty good. He said, this spot that they found is in my smell center. You know, they've laid out the map of the brain a little bit. So then he got really worried. I'm smelling crazy stuff. And so he went back and they said, you know, even if you say this, we are not worried, we don't want to scan it. You scan me again. And they fight. They did. Found the tumor, had open surgery. And he's doing well. He's finished his studies in the meantime, doing a PhD. So here's that, you know, you can read that at home. So then he decided, you know what, he was ill, of course, after this operation, and he said, you know, I'm bound to the home. You know what, I'm a very good student, so I'm going to study this tumor. Just, you know, to use the energy and the frustration. It's going to give me the data. I'll study this stuff. And then, of course, as anybody else asking these both questions, the data comes. Okay, they were very kind. They worked with him. Normally, if you I'll tell you another story from this country in a moment. So they gave him the CDs. Normally they say, no, those CDs are ours. They're not yours. You won't get them. But this is my tumor. It's our data. Sorry. That's the first disappointment you can run into. So, but of course, the tools to use it weren't there. So how useful is it? all kinds of legal. They said we can't send it, maybe we will send it. So basically, just accessing your own data, your own medical data, is a big issue in this modern world where we talk about big data routinely, right? And so this guy became a bit frustrated and started shouting out to this was snippets from newspaper articles, you know. And he says, you know, why is there no Google Maps of the brain in the meantime? What is this, you know? Why are cars much better developed than, you know, maybe more important things when it comes to life? And so, no man's land still, right? People are moving, a lot of legislation that impedes all of this, but then ethical considerations have to be taken into account, of course. So these are big issues, but we're in, you know, in early days of all this. So that has to change. Another person, a Dutchman, used to hammer this story home even more, actually. Yeah? And Dirk-Jan van der Pol, this was a text for the Health RI conference, the Health Research Infrastructure, you know, making all this technology and computation, putting together to do something in the big data in medicine. So he would talk about his problem, what is his story. It's also a cancer story, another complex disease, but he had thymus cancer. Some organs somewhere here. That was removed, and he was ill, but he could lead his life. And then, a few years later, he developed a bad form of COPD, the lung condition, restrictions in the lung. And that's normally a family thing, it's genetical. And so he went to his GP and he asked, hey, you know, the whole family doesn't happen. Is there a link between me getting the thymus extraction or the thymus cancer and that I contracted COPD? And, you know, the GP knew about Medline and he went on it and he didn't find anything and he should have said that, but he told them, no, there is no connection. That's what doctors do. When they can't find it, they say it's not there, right? And that's sometimes not a healthy way of looking at stuff. But anyway, we already had looked, of course, in Medline and he knew that it was difficult, so no answer. Then, you know, fortuitously he came onto an American sort of chat site, and there were specialists connected to that. And although he was already rich because he was an early day data science, so he knew about data in general, he liked it that it was free. He got on the chat with a specialist, and this, now I say something that is also data that doesn't sound like data. So the specialist said, interesting. Let me get back to you in 15 minutes. Okay, he waited. Specialist came back in 15 minutes and he said, I checked Medline. I couldn't find any connection. Then he knew. But then the specialist said something very interesting. But I'm intrigued, he said, because I get this question a lot. Is that data? We don't know anything, but it's a frequently raised question. Does that add to the importance? Maybe it does. Are those kind of things used in science? No, they aren't. Should they be used? Yes. How can we do that? Internet, chat forums, whatever. So there's a lot we can, you know, failed experiments are normally lost. You know, oh, I don't want to know about this in the waste bin. No. You know, you need to use data as it comes in a much better way than we do so far. Okay, so he decided, he was still ill, you know what, if there is question is there, I'm a data scientist, I can't work because of the disease, like just the other student, I'll spend my time in cracking this thing and establish this connection if there is something or not. And you know, well, I don't even show you his frustration because it was even higher because there's hospitals in the Netherlands said, look, we don't give you the data because we don't feel like it. Oh, okay. That's very nice, thank you very much. And but then, you know, he persisted, he got some stuff, he used public data and started data wrangling ever after, you know, putting one format into another and trying his stuff. And indeed, beauty is, he became a world expert at the link between thymus extraction, thymus cancer, and COPD. World expert, just a citizen. So we also talk about citizen science now. Intelligent people asking intelligent questions. There should be an infrastructure to make that happen. You know, do you need, you know, you need a driver's license, admittedly, for driving a car, but any special course to understand the engine? In the beginning days, every car driver was an engineer. The thing would break down every 5 minutes or so. That doesn't happen anymore. Technology is gone ahead. So should we go in that situation? And you know, some people, and part of them say, yes, basically, you need to make an infrastructure that allows no data wrangling, but just asking, combining data, being able to ask intelligent questions with good analytic software. Okay, so maybe this is the point to break. A few more slides after the break. It's 48 packs. You deserve a coffee. Could you still get me in the back? Good enough? All right, thanks.